Hey guys, today I'm going to go over cDNA library production and lesser known aspects of cloning. This is intended for people who already have some experience with cloning. I realize that I really have a need in these videos for more figures to help you better understand what I'm talking about, so I'm going to be ordering a tablet so I can draw things out for you. The goal is to, in a simple way, show you instead of tell you what I think is really interesting in biochemistry. cDNA library production would be a great topic to draw out for you, but I'm just going to explain it this time. Making a cDNA library requires a starting point of mRNA. So you have a sample of mRNA that you want to convert to cDNA so that you can do some manipulation of that cDNA or do expression of that cDNA. And this can be really useful for expressing eukaryotic genes in bacteria because the mRNA sample likely has a lot of spliced mRNA where the introns are removed. And so once you convert to cDNA, you now have a DNA sequence without those introns that you can express in bacteria, because bacteria won't be able to have the capability to remove those introns once they're transcribed to mRNA. So you take that mRNA sample and you have a poly T primer that you anneal to the poly A tail of the mRNA, and then you have a reverse transcriptase that will then extend from that primer to the 5' untranslated region of the mRNA. So you go all the way to the very beginning, the 5' end of the mRNA. And you've now made your first complete DNA strand that's complementary to the mRNA. And you have a fully hybrid duplex with mRNA and DNA. Next, you use RNase H to degrade the RNA in that duplex. And then you extend off those RNA fragments with a DNA polymerase. And if you use a DNA polymerase such as E. coli Pol1, you can actually create an entire continuous second strand that doesn't have any nicks in it through nick translation, because that polymerase, that E. coli polymerase, has both a 5' to 3' elongation activity and a 5' to 3' exonuclease activity. After this, you have like double-stranded cDNA, essentially. You've made your cDNA and you want to amplify that DNA in a cell, you want to transform it, or potentially you could go directly to expression, but typically you want to amplify in my experience, you want to amplify the DNA, purify a large amount of that, and then go to the protein expression part of your experiment. But your workflow may totally differ depending on what you want to accomplish. So you have that double-stranded cDNA fragment and you want to do some A-tailing so that you can put it into a vector, and we're just going to put it into a circular plasmid vector. And so you'll do a 3' tailing on both strands, and you can use DC, use an enzyme called terminal transferase. And if you just put in that DCTP nucleotide with that double-stranded cDNA, you'll make a 3' tail on both sides with DC. And then for your vector construct, you'll add a complementary base. So in this case, it'd be a DG 3' tail in a separate reaction. Then you combine the two, and since they're complementary, you should form a circular plasmid that has one copy of the cDNA and one copy of the construct. And you don't even have to do a ligation. You can just simply anneal the two and do a transformation, and then the ligation will happen in E. coli. And also DNA Pol1 will replace any of the RNA in that plasmid in the cDNA portion. It will convert that RNA to DNA. Never made a cDNA library, but now I feel equipped mentally and understand what's happening so that in the future, potentially, I know what is possible and could figure out how to make a cDNA library and study mRNA more closely. More generally, once you do a transformation of a plasmid into a bacteria, such as E. coli, you can do a plaque hybridization to identify a target of interest. So this will hybridize to the mRNA that is transcribed. And so you'll have a probe that is complementary to mRNA with a dye attached to it. And then you incubate this with your agar plate, and you'll see the colonies that have expressed that gene will light up as a certain color on your agar plate full of little bacterial colonies. Another option is doing antibody detection. So you have a labeled antibody that has an affinity to a target protein, and you wash your plate with that antibody, and it should stick to any of the colonies that are expressing a particular gene of interest. And you can do just a single antibody, or you can do a mixture of antibodies of interest. antibodies against proteins of interest. If you want to do experiments to kind of show a connection between function and DNA sequence, you can screen binding or enzyme activity using phage display. This is where you manipulate the DNA of a bacteriophage to fuse the protein of interest to the coat protein. And once it's expressed, that coat protein will display that protein of interest on the outer surface of the bacteriophage. One tricky way of going about making this kind of construct is to use this phage mid vector has an origin of replication for plasmids, so for a circular plasmid, and also an origin of replication for phages, bacteriophages. This allows the vector to be packaged into a bacteriophage, and it can also be replicated in a bacteria like a plasmid would. So you can amplify the DNA in bacteria for genetic manipulation. And then you can go back at the end to package it into a bacteriophage for your binding study, or enzymatic studies. You can know that you have created the right bacteriophage construct by using labeled antibodies that I mentioned earlier that could be used for colony identification. But you can also use them for confirmation of your bacteriophage construct by using labeled antibodies to identify the plaques resulting from phage-induced lysis of cells. The plaques can be blotted onto nitrocellulose and then treated with the antibody to identify plaques that have expressed a protein of interest. Often when you're creating a construct, you use PCR, and if you're making point mutations, then you'll use primers that are not 100% complementary to the template because they have the changes that you want to incorporate into the gene. PCR has limitations. It can only be used for genetic fragments up to 40 kilobases, and it can only tolerate a certain number of mismatches. However, the stringency of the hybridization can be changed by altering the conditions of the hybridization during PCR. You can change the temperature, so an increased temperature would lower the stringency of the PCR. So maybe a lower temperature would be beneficial if you wanted to make an increased number of nucleotide changes. Organic solvent also favors denaturation, so adding organic solvent lowers the stringency PCR. Low salt also lowers the stringency by causing denaturation due to a lack of shielding between the negatively charged phosphate backbones of a duplex. A lot of scientists use a lac promoter, and this is part of the bacteriophage lambda system. This promoter is often a little leaky even when the repressor protein expressed from the LAC-I gene is present. Often the natural production of this repressor isn't often enough, so sometimes scientists will introduce a LAC-I repressor on another plasmid to lower that leakiness resulting from there not strong enough binding or there not being enough repressor to block transcription for target gene. But sometimes it's not always necessary. Leaky can be okay. Other ways to get around this leakiness, one way to increase the strength of the promoter is to make a hybrid promoter with the tryptophan operon. So this TRP promoter is 35 base pairs upstream of the start codon, whereas the LAC operon is 10 bases upstream. So it has two promoters. The TRP makes it a very strong promoter, and the LAC promoter makes it controllable. So this hybrid promoter is both a strong promoter and useful for expressing large amounts of protein. If you want a finer control of your expression, you can use the ARA promoter, which is the promoter for the arabinose biosynthesis operon. And when you use low concentrations of arabinose, you get low levels of expression. But once you increase the concentration up to 0.02, you get increases in the expression. Another strategy that has been used for inducing expression A operon and initiating transcription is to use a temperature-sensitive repressor for the LAC operon. And this allows you to control the induction by a temperature change. If you increase the temperature, the repressor is no longer favored to be bound to the promoter, and it dissociates and transcription is initiated. I hope you learned something about cDNA cloning and other aspects of cloning in this episode. Have a great week.